I really appreciate your kind introduction. And I also want to thank Paul Parvis for inviting me. It's a great honor for me to be here. And one of the highlights of my day was having a tour with Mr. Parvis of your beautiful campus. I'm really impressed. I really love all the opportunities, and I saw, I'm sure, only a small fraction of them. So thank you for hosting me. I really appreciate the opportunity to tell you about my work, and I'm hoping that you're interested and you'll have some questions, and I'll be happy to entertain those. So I wanted to tell you a little bit about this by way of introduction before I plunge right in. This is a conservation project, and you're wondering probably why the Hudson, why these plants, what are intertidal plants. I'll be answering all that along the way, but the big point is that it's a chance to focus in depth on a facet of our flora that has not been getting any attention, and it certainly deserves that attention. So what I'd like to do is just spend a little bit of time talking about the Hudson River. The Hudson River, you probably realize all this, is a tremendously important river, whether it be culturally, historically, or commercially. So just consider culturally, for example, that it's been the target of the Hudson River School of Painters, so prominent in the history of American art in the 1800s. And then commercially, it's a navigable river for well over 150 miles. Just think also commercially about some of the important places along the Hudson. Albany, the state capital, is right on the Hudson River, and that's why it's there. So historically, think about West Point, so prominent in the history of the American Revolution and onward to, and continuing to today. West Point is there because of the Hudson River. So I could go on just that. That's not the point of this. The point of my subject in my lecture tonight is to talk about conservation. And I want to put this in a broad framework. So if we think about our flora here in northeastern North America, from a conservation standpoint, we realize that the plant species fall into one of three groups. And the first one, exemplified by plants like this cardinal flower, are native plants whose populations are relatively stable. A second group are non-natives, like the lesser celandine. These are usually expanding, and many of them have become invasive. I'll be talking about invasives later on. And then finally, we have a group of plants exemplified by the swamp pink that are natives that are in decline. And we actually have a serious problem with declines in our native flora, though this has been studied only a little and only very, very recently. So don't just take my word for it when I say our flora is in decline. Let me review the literature, and this is about all of it. So it's been relatively recent, you can see, and also few. And these studies are highly concentrated in the states of New York and Massachusetts. One holdout is Marion County, which is the county for Indianapolis. The point of this review of literature is that we see in all of these studies a decline in the native flora. And the proportion ranges from 10% in Indianapolis all the way to 44%. All of these are geographically concentrated local studies. I'll come back to that in just a moment. But think about what this leads to. The consequences of declines are extirpations. Extirpations are local disappearances. This means the loss of species, in this case the plant species. Ultimately, of course, these could lead to complete loss, in other words, extinctions. And what I've done here is to just take a few states, including those that we just saw on the last slide, and look at the percentage extirpation of native species, and it ranges from 2% to 15% in this sampling of states. So these two sources of data, extirpations and a review of the literature for local floristic studies, show us that we do have problems with a decline in our native flora. So this brings me to why I got into this project, the study of the intertidal plants of the Hudson River. Well, all of those studies that I reviewed were locality-based. They were geographically centered in local areas. What we really need now is a habitat-based approach. And that's what this is. This is the first habitat-centered approach to looking at the health of our native flora. Now, what is the intertidal zone? The intertidal zone is that stretch of land that's available for colonization by plants, as you see here, between the limit of low tide and the limit of high tide, right here at the tree line. And this is actually a healthy intertidal habitat. It's quite densely covered with plant life. This is an example of an intertidal plant species. So this is Sagittarius subulata. It's a member of the genus that includes plants called arrowheads. These are non-woody plants, they're herbs. And this one is restricted to the intertidal zone. So here, of course, is one individual. Here is a number of them. And here, in this zone, low in the intertidal zone, and this is extreme low tide at this site, here you can see quite a large population of them. So this is just one example. That's an intertidal plant. And many of these plants that live in the intertidal zone are restricted or nearly restricted to the intertidal zone. So why study them? I already mentioned this. Many of them are very poorly known, even to botanists. So what I did, and I admit this is fairly unscientific, but it gives us a quick piece of data to look at to back up what I was just telling you. I did a little survey among professional botanists who spent a lot of time in the field in the New York region. I actually checked with botanists in New York, New Jersey, Pennsylvania, and Delaware. And I just asked them simply, have you seen this plant, Liliopsis chinensis? Liliopsis chinensis is a member of the carrot family. Chinensis means from China. You're beginning to wonder, wait, I thought these were native plants. They are. This was a misnomer. When it was described by Linnaeus, he thought the specimen was sent into him from China. So there was a mix-up on the labeling. It's actually native to Eastern North America, and that's it in terms of its entire global distribution. So I asked, have you seen Liliopsis chinensis? Many of them said, I've never even heard of it. But only 32% of them said, yes, I had seen it. These are professional botanists whose jobs include field work. Many of them have as their sole job tracking the health of native plants. And I surveyed 31 individuals. Okay, next, what we do know about these plants, the little we know, indicates that several of the species are restricted to the intertidal habitat, and several of them are restricted to northeastern North America. We call this endemic. They're endemic to northeastern North America. That means their total global distribution is in this area. And this is one, this bidentoides, is one example, and this is actually one of the more common of these intertidal plants. So this plant is a member of the sunflower family, and its total global distribution, as I say, is here, but even more, realize it's limited to the drainages of only 7 rivers. You begin to see the restrictiveness. Couple that with the fact that even on these rivers, the intertidal habitats are rare. So on the Hudson River, for example, many places, steep rocky banks come directly down to the river. There's no chance there for intertidal plants to grow. Most of them live on the mud flats that gradually slope into the river. Not so here. Also along the Hudson River, on both east and west sides, we have railroads, and these steep, rocky banks that humans built for the railroad eliminate the possibility of intertidal plants growing. So the habitat's rare. The habitat's extreme. When you're a plant, this is pretty extreme. And what do I mean by that? A whole list of things. First of all, the tides. So every day, within a 24-hour period, a plant is covered, it's inundated by high tide, and then it's exposed by low tide, and the whole thing, that cycle, is repeated. Every day we get 2 high tides and 2 low tides. Most plants just can't deal with that. That alone is extreme. But we also have fluctuating salinity. The reason why the river's tidal, of course, is because it's connected to the ocean. Well, that salinity with the ebb and flow of the tides moves up and down the river, but also we have a seasonal fluctuation. Usually we get drier weather later in the summer and the salt front pushes farther up the river. Storm events affect the salinity too. Turbidity. Storm events wash a lot of sediment down from the uplands, especially in cleared agricultural fields. That leads to sedimentation. Also, wave action, particularly when we have ships going by, so we contribute to that. And then all of these together mean that the substrate is tremendously dynamic. A given spot any one time will not be there in a few decades, or maybe less, because of all of these factors. Now, I'd like to introduce the Hudson River Estuary for you and tell you a little bit, because I don't expect that everyone is intimately familiar with it. It ranges from the southern tip of Manhattan at Battery Park all the way north of Albany to the city of Troy. It's long. This is a tremendously long estuary. And I've given you comparisons here with other great estuaries in the Northeast, the Delaware River, the Potomac, and the Connecticut. The Connecticut River's 60 miles long. That's a great estuary. 153 miles. When I say estuary, I mean the tidal part of the river. 153 miles north of Albany. In fact, the federal dam at Troy is what stops the river from being tidal. The dam was built way back in the 1800s. Before the dam, the tidal reach of the river was even greater. The tidal amplitude is moderate. As far as tides go, this is actually quite modest. If you're familiar with other areas like in New Brunswick, the Bay of Fundy, where the tidal amplitude can be over 30 feet, this is very modest. And then another point is that the Hudson's very unusual in that the tidal habitats are almost completely limited to the river itself, because the surrounding areas are uplands, many times highlands. And so tributaries are not at all tidal, or if they're tidal, for a very short distance, tenths of miles at most. I want to come back to salinity, since I know that many of you are interested in biology. I thought you'd relate to this. The field of biogeography is an important one, and it's the study of why organisms occur where they do in our natural environment. And I have an example here of the genus Bulbochinus. These are herbaceous plants in the sedge family. And the three species I'll be showing you are all close relatives. And what I'm doing is I'm mapping their distributions for you. Bulbochinus robustus is a salt-loving species. In fact, I'm just mapping the Hudson distribution. This is a widespread plant that occurs all along our coast. So actually, its range is along Long Island in salt marshes. Historically, it was probably in this area too, but we developed those areas so early that we did not yet collect specimens from them to document. So I map only those occurrences for which we have specimens to prove their former existence or current existence. Well, let's take the next species, Bulbochinus novianglii, the New England Bulbochinus. And you can see there's a slight overlap in distribution with robustus, the first species, but this one is restricted to the brackish water. That's the area where there's a mixing of fresh and salt. And then finally, Bulbochinus fluvialis meaning riverine, the river Bulbochinus, that one occurs all the way up to Troy and this far south. There's actually no overlap, though close, with Bulbochinus novianglii. This one's restricted to fresh water. So these organisms partition the salinity gradient quite specifically. Many of the species I'll be showing you are a bit more generalist, but many of them are quite specific like this. It depends on the species, its own tolerance for salt conditions. But this exemplifies the importance of salinity in the environment. Another thing, I made the point about the environment being extreme, but another thing that makes this interesting is that extreme environments select for extreme adaptations. And we've not studied these at all. just think of what a treasure trove this is. We take our cue from nature and development of new technologies. Here's an opportunity. So when I was out there, I got thinking about this when I was doing field work, and I think I found one of these extreme adaptations. It's completely unknown. It's not in the literature. I found this. I may be crazy when I tell you what I think's going on, but I leave it to future botanists to study this. So this is that same species I showed you earlier, Sagittarius subulata. And if you look closely, especially in this picture, I hope you can see what I did was I photographed through the water of the rising tide. And these flowers captured, each one captured a bubble. Can you see that bubble in there? Okay. So this is not happenstance. It's not just this site. This Sagittarius subulata is pretty common, and what I did was I looked for it once I caught on to this, and it's a regular event. It may simply be a coincidence of the physics of water in those flexible petals, but it may actually be adaptive, and this is what I suspect is going on. This is my hypothesis. You know the life cycle of flowering plants. Pollen is deposited on the stigmas, and then what it does is it grows a pollen tube, which then can allow for fertilization, and then of course seed development and fruit development and completion of the life cycle. Well, it takes time to grow that pollen tube. If this bubble were not there, I suspect the action of the water could wash away the pollen before the pollen tube had time to grow. So I hypothesize that this bubble is allowing more time, it's buying time, for the pollen to grow the pollen tubes to allow for fertilization. Maybe a crazy idea, but it should be studied. It's interesting nevertheless. Okay, finally, and this brings me to the overall point, is that the intertidal habitats are facing great threats. We are the cause of many of these threats. We develop intertidal habitats, we pollute them. We are also, through our actions, leading to, apparently, I realize this is controversial, but we do know that climate is changing, that our environment is warming. We are apparently the cause of that. That's what's controversial, but it is a fact that global temperatures are rising. Well, with that, the sea level is rising, and that salt front will be pushed inward. We call this northward, upriver. We call this saltwater intrusion. What effect will this have on the plants? So, given the restrictiveness of these intertidal plants and the threats they're facing, we expect that these plants are very sensitive indicators of environmental change, and it makes them among the best to study for a habitat-centered approach to native plant conservation. That's why I chose these intertidal plants. And given that I work at the New York Botanical Garden, what better thing to do than to study the Hudson River? Such a great river with such a great range of intertidal habitats. So my hypothesis is that these plant species that are intertidal, and when I say that I mean restricted or nearly restricted to these intertidal habitats, they are at risk of extirpation within the near future. And they should be conservation priorities. They're not now, I'm sad to say. And one of my goals is to make a change, see that happen. So my objective is to test this hypothesis by doing a field-based study that's historically grounded. And I'll be explaining what I mean by that in just a moment. So what I did, in terms of how I have done this study, is I have first had to go out and compile a list. We don't even know how many species of plants are restricted to the intertidal zone. You have to start somewhere. So I began by compiling the list. And I did this by reviewing the literature, by searching the herbarium. The herbarium is a museum of plant specimens. And we have the Western Hemisphere's largest herbarium at the New York Botanical Garden, fortunately. So that provided an opportunity. But then also I built upon my past experiences in intertidal habitats on the Delaware River and on the Chesapeake Bay and its tributaries. One thing I want to make clear is that many species, over 300 of flowering plants, occur in intertidal zones. Only a minority of them are restricted to the intertidal zone. So when I say intertidal plants, I mean these plants that are restricted. Some of the plants that occur in the intertidal zone that are not restricted are pickerelweed and cardinal flower. These are generalized marsh inhabitants. So you can find them well inland in non-tidal habitats. They occur in the intertidal zone, but they're not my focus. My focus on these few that are so sensitive and restricted. Okay, the next thing I did was this historic component. I assembled a historic baseline by studying the herbarium specimens. The herbarium specimens provide this tremendous opportunity because they are the physical proof that a certain plant species occurred where it was collected. And to do this, I worked at the New York Botanical Garden, but also 7 other herbaria that have great collections of intertidal plants. In total, I studied 683 specimens to build this baseline, and the collection record extends pretty continuously from the late 1860s up through the present, essentially. Actually, until I started this project, the most recent specimen I found was 2003. There was one earlier collection that was undated, and we estimate it was collected about 1825. Well, we need to know the quirks, the good, bad, and the ugly of the historic record. First, you want to realize that these specimens were collected mostly opportunistically. People visited the intertidal habitat when they wanted to, for the most part. The only exception was in the 1930s. So what I'm showing you is the 2 most common species of intertidal plants in their collection record by the number of sites that were documented by decade. And in the 1930s, there was a bit of a concerted effort by a couple of botanists from Cornell University. They visited a nice set of sites, but it was in the teens. It was 13 to 15 sites that they visited. It was not a completely systematic survey. Otherwise, in all of those other decades, it was just hit or miss, even for these most common plants. So you can see relatively few collections per species, even though these are the most common. So let me show you a real specimen. There's the label, Isoletes riparium, the river quillwort. High water line, mouth of creek, just above Peekskill. August 1869, William Leggett, the collector. It doesn't get any better than this. And most of these specimens are like this. What do I mean? What I mean is, there's Peekskill. Mouth of creek above Peekskill. It can only be Ansville Creek. It's that easy. It's beautiful. Having this historic record, it's data waiting to be mined. It's botanical gold waiting to be mined right there in the herbarium. Sad to say, I went to Ansville Creek, the mouth of Ansville Creek. I even went well up the creek. The habitat's still there, but the plant is not. So what these historic specimens do is they document 88 different sites of collection, and you can see they're pretty good coverage throughout the estuary. Then we did the fieldwork. Of course, that was the most fun. And what I did, and I really should say we, I have a team, I'll be introducing them to you in just a moment. We explored mostly these sites of historic collections, but we also squeezed in some sites that never before had been studied. And then every day we were constrained by the tide, so we had this narrow window of opportunity and we made the most of it. Basically it's 4 hours, 2 hours before and 2 hours after extreme low tide. Any more than that, any more than 2 hours, and the waters are high enough that they're obscuring the plants. What we do the rest of the day is we scout the access points for the next day's fieldwork. Here's my team, Suniti Jog from the Nature Conservancy, Sarah Walker, a herbarium assistant from New York Botanical Garden. These were volunteers. David Warrior, president of the New York Flora Association. Fortunately, my grant funding allowed enough that I could hire David on a part-time basis to be my field assistant. And then Charlie Zimmerman, also a volunteer, like the first 2. He's also a herbarium curatorial assistant at the New York Botanical Garden. These folks are great, and I like to publicly acknowledge them because without their help, I wouldn't be able to do it. Many days I'm out there by myself, but most days they're with me, and I really appreciate their help. So what we've been able to do in the first 2 years is explore 51 sites. I have 1 more year of fieldwork. I project 1 more year of fieldwork left. And realize everything I'll be telling you in terms of results will be preliminary, necessarily tentative, because one could say, well, maybe you'll find it in those sites that remain. But I'll leave that. The other thing I should mention is that just because of the way it's worked, I've not spent much time in the mid-estuary yet. But there is a reason for that, and that is the fewest intertidal sites are in there, because remember that view I showed you of the rocky banks coming right down to the river? Most of that stretch of the river is like that. There really are not that many intertidal habitats. Okay, so what is it like? I want to give you a sense of this fieldwork, since I said that was the most fun part of it anyway. Well, what we do is we use kayaks. We put in where we can. Access is not always easy. But we put in where we can, we paddle to our site, and then we get out, and we just explore very intensively. We try to document the presence. That's easy, but even more challenging is the absence. If we say something's not there, we want to be sure of it. So we're very thorough when we explore these sites. Okay, I want to give you a real example of what it's like to hunt for one of the members of the intertidal flora. It's one that I mentioned to you before. So this is a real example. This one day I was near Coxsackie in Albany County, southern Albany County, so it's a little bit south of Albany. And I was on the west side of the river, and the habitats were not very fruitful. The habitats were there, but I wasn't finding the plants. And I still had some low tide left. So I paddled across the river to Rensselaer County, which has been very little explored. And I thought, oh, this will be great. I can go to some place that no one has ever visited. When I got there, I was disappointed because I saw this timber bulkheading, and then I saw this rock piled up by humans that covered about half of the intertidal zone. But I made the effort to get over there, and after all, I didn't have much time left in low tide, so I said I better get out and start exploring. So this is the view when I turned my back. I turned 90 degrees, and I faced inland. So again, you can see this rock pile here, but the upper half of the intertidal zone was intact. So I want to focus you, whoops, I'm sorry. I want to focus your attention to the center, and there it is. And what was it? It was the river quillwort. And this was a day for celebration. And I'll explain that. I'll leave that for now. I'll leave a tease for you. So this is what it's like. This is a self-portrait. It's wet and it's muddy, but when we do this work, we don't mind that because all of us are quite passionate about plants, and this is ending up being a very interesting project. So we're motivated by conservation. We really want to know. We want to do our best. I want to give you a sense of what else goes on out there. You can make out the dim outline of this creature, and I have to tell you, when I'm in the field, it's very rare that I feel fear. I'm very comfortable in the field. I love field work, but this day I felt fear because this was the largest snapping turtle I'd ever seen. It was a good size one. You're in the ballpark of the shell size. The head was this wide. Now, that wasn't scary. What was scary was the thing was coming toward me. When I'm in the field and I see animals, usually they go away. And the way this worked out was I was in a bay that was U-shaped. And I had already explored all of it, and I wanted to get back to where my kayak was, so I wanted a shortcut. So I was wading up to my thighs in the water. So I'm splashing, and I'm already about halfway in fairly deep water, and I see this thing. And I just stopped, and it came toward me, and I slashed my foot a little bit. It kept coming toward me, so I got out of there really fast. Another thing that we have to be aware of, as I mentioned before, is the Hudson River is an active shipping channel. This is an ocean-going, international-bound freighter. This is not that uncommon. These things move really fast. Tugboats are much more common. They're smaller, they move slower, but still, you have to be careful, especially when you're crossing the river. Not just because you don't want to be in front of one of those things, but the waves that they leave behind upset your kayak if you're not careful. I haven't had that happen, but I don't want to find out. And then my own stupidity. That's what I have to fear the most. You can probably guess what happened here. It was low tide. It was low tide. I brought up my kayak. I got absorbed in my work. And before I knew it, I said, wow, that's a nice kayak. Wait a minute, that's mine. Well, fortunately, that was a day that David was out, and he was able to go out and retrieve my kayak. But my pride, of course, is what really suffered. Okay, back to business. So the last part is analyzing the data. And at its basics, what I'm doing is I'm comparing the historical record with the current. But I do that by georeferencing, by getting the latitude and longitude for all of these collections, all the historic ones as well as all the current ones. I do the current ones in the field when I'm doing the field work with a handheld GPS unit. And then what I use is GIS, or Geographic Information Systems, to plot and analyze these data. Okay, what are the results? Well, first of all, in compiling my list, I came up with 21 species of plants restricted and nearly restricted to the Hudson River Intertidal zones. Then the conservation status. So I broke the status of the species down into 3 categories. First, those species that are secure. These are the ones for which the future is bright. They're doing fine. And what I did is my criteria is I set them at more than 15 sites, more than 10,000 plants in total that I and my teammates have been able to find. Imperiled species are those that are in trouble. They're in decline. Moderately imperiled are those that have 5 to 15 current sites and or 1,000 to 10,000 plants. Critically imperiled are ones that are in more serious trouble. These are the ones that have fewer than 5 sites, fewer than 1,000 plants. And then sadly, a third category, those that are extirpated, those that are already gone, those that we knew were there from the historic specimens, but they're gone now. Okay, so an example of a secure species, you've been seeing this one, Sagittarius subulata. And what I do here is map the distributions, and my key is the historic sites are these solid red circles, and the current day sites are these open circles with a dot in them. And as you can see, both historically and present day, the known populations span the estuary. This is the most general of the species. It doesn't seem to have a problem with variations in salinity, unlike the example I showed you earlier. Another secure species and another generalist, and those probably go hand in hand, the health of the species and the fact it's a generalist, is Amaranthus canopinus. This one we know 21 populations, again distributed mostly throughout the estuary. Now those that are in decline, those that are in trouble. Bulbochinus noviangli, that one that I presented to you earlier as a brackish water requiring species. So already it's ecologically quite restricted, neither fresh nor salt, but in the mixing zone. This one, we know only 5 populations in about 1,100 plants. And you can see they're tightly clustered. That's the only place in the whole estuary where we know this species. Okay, the river quillwort. This is one that's in serious trouble. It was known from a number of populations historically, but that's not the case. I gave you the tease earlier, and I said, when I found it, it was a day for celebration. Well, you know already why, because it's quite rare, but even more, when I found it, this was the first time it had been seen on the Hudson in over 70 years. So I don't want you to get the idea that it's all gloom and doom. This is admittedly a fairly short plant. It's about that tall, just maybe about 20 centimeters. It's not the easiest to find, but other botanists had been out there looking for it. Did it come back? Is it just hard to find? Did I get lucky? Maybe it's all of those things. I don't know. I can't answer those questions. But what I will say is this. I suspect it's been there all this time. These plants are so sensitive, it's unlikely it came back. It certainly didn't come back from the dead. We don't know that. My job with this project is to document the patterns. I'll be speaking in just a few minutes as to possible explanations, but my first job is to document the patterns. So I don't want you to get the idea from this one example that all of these species that are in decline will somehow bounce back. I don't call 129 plants bouncing back. This plant is teetering on the brink of extirpation on the Hudson. And what thrills me is we still have it, so we still have an opportunity to make a difference. And maybe we can see this come back to where it rebuilds to healthy populations. Sadly, we do have this third category of extirpated species. And this is the most egregious example. Parker's pipewort, Areocolon parkeri, once was common and widespread throughout the Hudson. It was known from 14 different places. These are the vast majority of sites that have been visited by those botanists from Cornell in the 1930s, and they're the ones who collected most of these specimens. The last time it was seen was 1944, so this brings me back to the point I was making just a bit ago. We can have all the wishful thinking we would like maybe next year I'll find it, but it's time to get realistic. Conservation, effective conservation, is not built on wishful thinking. It's built on reality. And it's unlikely, I will never say never, but it's unlikely we will find this plant, given that we've visited 51 sites in total, and all but one of those 14 historic sites. I think this one is likely gone. It's an example of, it turns out, 5 species that are extirpated, apparently extirpated from the Hudson. So the overall summary, again preliminary, because I have one more year of fieldwork, but for now, the overall summary is this, that only 29% of the 21 species are okay, are doing well, maintaining their own at the least. The vast majority, almost half of them, are those that are in decline. And then almost 1/4 are gone already, 5 of the 21 species. So the results are grim, indeed. Why? I mentioned to you that I would come to this. Why? I don't know. All I can do is suggest some possibilities, and the possibilities are numerous. I suspect it's a combination of these factors, some being more important than others. Habitat destruction, we would think outright habitat destruction, like through development, would be a leading cause, but I'll be honest with you, that seems to be the least likely, because most of the development happened in the past. It's already happened. Very few intertidal sites are under threat now from development because of the laws that we have in place to protect them. That's a good thing. Erosion, especially from big ships making all those waves, that's a possibility. I don't think it's one of the strongest ones. Sedimentation, that's certainly a possibility. When silt is deposited on these intertidal habitats, it cuts down on photosynthesis. If it's extreme, it actually buries the plants. Dredging, what does that do? It actually causes more severe saltwater intrusion. We need to dredge, but sometimes the dredging causes unintended consequences by making that saltwater front move farther upriver. Pollution, I'll be talking about this in just a minute. I hypothesize that one form of pollution is one of the leading causes for these declines. I'll explain in just a minute. So that is one of my top 2 suspected causes. The other one is the influence of invasive species. So invasive plant species are a major and growing problem on the Hudson River, as they are in almost all ecosystems in the Northeast. One that has been with us but continues to spread is Phragmites. That is a huge wrasse. It gets up to 15 feet tall. You can see my kayak for scale. And what it does is it creates a dense monoculture, causing so much shade that these intertidal plants cannot survive. They're also just literally crowded out. Their space is taken by the Phragmites. These others are recent invaders. They're all members of the sedge family. I found all of them last year, and 2 of them are new to the state of New York. So it's suggesting that the Hudson River may be a place where a lot is happening with invasive species. This first one had been documented once before in New York State from Long Island. I found it farther north. This next one is new to New York. It was known from adjacent Massachusetts, so it's not a huge surprise it would be found here. And then this last one is quite a bit farther north than it had ever been found, and it's new to New York. This is the one of these 3 recent discoveries. This is the one that is the most problematic. It is invasive. It is presently crowding out native intertidal plants, including some that are very sensitive in an otherwise very high quality environment. So I'm hoping to work with people in the New York State DEC, Department of Environmental Control, and have them work on a way to effectively manage the invasive problem there at that site. Now on to the pollution. To cut to the chase, I posit that excess nutrient pollution is the leading cause for the decline of our intertidal flora. Historically, the intertidal zone was oligotrophic. It had few nutrients. Now it's eutrophic. And if you need to be convinced of that, there are many ways of doing it. I show you one. This is a bay, and it's filled with the invasive water chestnut, all of this stuff on the surface. But what's in the water is also choked with other species, many of them exotic too, or invasive. And I think it no coincidence that this particular bay is surrounded by homes with septic systems, some of which are probably leaking. But the excess nutrients flow down by way of excess fertilizer from farm fields, from sewage that continues to be dumped directly into the river in this year, 2013. So here's one of the results. This is a place at a different site, and I've seen this numerous places now. This is a place that used to be an intertidal habitat. It is still intertidal, but it used to host intertidal species. What does it host now? An overgrowth of algae. And I'm trying to get this identified. I don't know if this alga is even native. Why does all of this matter? I'm nearing the end now. Well, this is the first modern comprehensive study of the Hudson River's intertidal plants. And as I've already explained, these plants are special. They are worth conserving for their own sake, but also for what we can get from them from a selfish perspective. They are rare, they're limited to a very special environment, a unique environment, an extreme environment. Think of these adaptations that we haven't even discovered yet that these plants possess. Even more to the point of the selfish perspective, these plants provide ecosystem services. I'm from New York, of course. All I have to do to convince people of this is mention one word, and that word is sandy. When we have intact intertidal mudflats and other intertidal zones, those zones held in place more or less, they're dynamic, but they're more stable when they're vegetated. When they're there, they buffer the effects of storms and storm surges. Sure, they're not going to stop a 15-foot storm surge that slammed into the southern tip of Manhattan, but that storm surge surged up the Hudson River and menaced many other communities, even at only 2 to 3 feet, where even higher, 5 feet. These intertidal zones break that surge, they reduce its energy, they help dissipate it. That's just one ecosystem service that these plants render. Also, as I mentioned, these are indicators, they're sensitive. We need to wake up. We've had so many other indicators. This is not really news, but we should be working on the Hudson River. And that's the positive outcome of this, is that these plants will help us in restoring our environment. And that's what I'd like to talk about right at the end, is some practical applications. First of all, New York State does have some laws that protect native plants. And what I found is that what's on the books is out of line with reality. And I'll explain just a few here as an example. So I have the New York Natural Heritage Program listing, which is tied to the law. And it's this code that S1 species are known from 1 to 5 sites, S2, 6 to 20, and S3, 21 to 35. All of these are protected. Well, Biden's Hyperborea, Cardamine longii, both of those are on the list. They're both based on misidentifications. I had to look at the specimens because they were reported from the intertidal zone. They're not there. So in a way, that's a good thing for us. Lilioxys chinensis. Well, it's even rarer than we expected, so it needs to be S1 instead of S2. That's kind of the theme here. But in this case, we can actually go the opposite direction. We can downgrade. So instead of S1 to S2. Here's one going from S2 to S3. And then in this case, this is sort of our celebrity for the show. I've been talking about Sagittarius subulate. It's actually common enough, we should remove it from the list. Again, good news. There is some good news in this story. And then another point I want to make is that what we've learned about these sites can guide us because resources are always limited and limiting. We have to put our energy and our money into those places that most deserve it and can most benefit from it. So historically, what were the most, historically, what were the most important sites? Rogers Island on down to Iona hosted between 10 and 13 intertidal species. These were the best of the best. Nowadays, it's diminished, but we still have some great sites. And just the fact of identifying these sites allows us to say we need to conserve these places particularly. And fortunately, some of these are already protected areas. So by this point, I hope that I've convinced you that these are special organisms growing in a very special environment. And I'd like to have some questions in just a moment, but I'd be remiss if I didn't thank all of those people who make this work possible. First of all, my funding comes from My Good Fund Trust and the Hudson River Foundation, and I have a couple of people who have been at, or and are currently at, New York Botanical Garden who helped me in securing this funding. Next, I have those herbaria that allowed me to go into their collections and study them. I have my field team. I also had a number of individuals who gave me access that made my work so much more efficient because I didn't have to paddle for hours to get to the site. And most of these were private individuals, landowners right on the Hudson, very generous people, but also some marina owners and managers. I had Charlie Zimmerman, who was not only my field, one of my field volunteers, but also helped me with some scientific work. I had help with the GIS from Becky Hurdy. The New York Natural Heritage Program did share its data on what it knew about the distributions of these plants. And then I had a generous person share her kayak when I had extra volunteers come out one day. And then, again, some of these folks that were so generous in giving me access, they brought us in and sheltered us and warmed us up when we were overcome by storms and other calamities along the way. So I thank you very much for your attention, and I'd be happy to answer questions. Yes?
I actually have two questions. But the first one is, how do they count for the increase in invasive species?
How do we count for the what of invasive species? The increase in invasive species. That's a great question. Almost everybody asks that who cares. And I'm glad you care. Well, if I understand your question correctly, we don't know, first of all. And if anybody tells you otherwise, they're being deceptive. It's under study, though. That's the good point. It appears that species become invasive because they're removed from their natural environment. They go to an environment where they can grow, but they're removed from the competition and the predators, mostly herbivores, that keep them in check in their native place. We don't know enough about competition, but that's probably at least as important as the herbivory. So basically, these species are taken out of their environment and there are constraints that keep them in check where they're native. And they go to this place that's favorable to them, and they don't have those checks on them, and they literally go wild. Does that answer your question?
Yeah.
Okay.
And the second one was, relative to other rivers, like, is the Hudson River relatively clean in terms of polluting?
All of, well, I shouldn't say all, but most of our large eastern rivers are heavily polluted. And there's some really scary stories I could tell you about a number of them. The Hudson has some portions that are doing pretty well, and is certainly doing better than it used to. I've talked to old timers, residents who still live on the Hudson, and they tell me the way it used to be. So although we still have serious problems, we are getting better. That's the good news. Now, comparing the Hudson with something like the Delaware, both rivers are doing better, fortunately. Some parts of the Hudson are doing better than the Delaware. Overall, because of the length of the Hudson, we have better populations of some of these rare plants. All of these rivers need much more attention. Okay? Yes?
Congratulations first of all on finding that species.
Thank you.
Only 7 years ago. That's amazing. But what are you doing now in terms of this habitat? Are you trying to bring it back? Have you found more in any other areas?
Well, that was late last year when we found those three. One thing I didn't mention is all of the field work happens late in the year because that is the time of the year when these plants can be identified. Many of them are not identifiable at all earlier. So we start mid-August and we go until frost. So I found those populations in September and October. So have I found more? I hope to this year. I'll certainly be on the lookout. I am working with folks in the state of New York, and when I finish the project, I've given them interim reports, but when I finish, I'll be delivering a final report, and I like these kind of opportunities. I'm talking to as many audiences as I can. So I'll be spreading the word in as many ways as I can, working with them. My job, as I say, is to do the research, is to document these patterns. I will do all I can to try to push it to the next level, but it's really other people who have the expertise in restoration and taking conservation to the next level. So that's, my hands are a bit tied because of all my other job duties. Yes?
How can we as high school and rising college students impact kind of conservation of these intertidal plants and the species, the new invasive species that arise?
That's an excellent question, and I'm thrilled that you are interested, that you want to become engaged. Well, many different ways. That's the good thing. Get involved. Well, one of the things is put pressure, appropriate pressure, on those in power, especially our legislators. And as I say, do it appropriately. So even before you try to do that, to become an activist, learn, educate yourself. You're doing that. And you're doing that not only by attending this lecture, but through your education here at Taft, your excellent education. Maybe you're an environmental science student, I'm guessing, because of your interest, I hope. But anyway, learn about the species, because when the legislators might push back, they might say, I have all these other priorities. They do. A lot of things are happening, many bad things. The economy is still in trouble. And they might say, well, you know, our economy has to be the priority. We have to get people jobs. That's true. But there won't be an economy, there won't be jobs if we don't provide for our future. And that's where you have to push back with facts. So learn, educate yourself as much as possible. Another thing to do is get involved in your local area where you live. Offer to manage invasive species. Offer to, and again, there are appropriate things. There are different ways of doing this, some better than others, some more effective than others. But offer, be generous, be a volunteer, be a scientist if you're so inclined. When you see projects like this, and there are a lot of scientists like myself who do field work who ask for volunteers, sometimes can even pay them. But if you have the ability to join a team to do a project like this, you may want to do that. I could go on and on and on, but there's an offering. Thank you. Thanks for your question. Yes?
Are herbivores starting to eat the invasive species?
Not yet. Not yet. That's a great question. And with time, we hope. Now, let me qualify that. Native herbivores that are already here, not really, no. But sometimes we bring in those organisms that were munching on those species in their native environment. We bring them in a way to try to control them. This is called biocontrol. It's one form of biocontrol. We have to be careful because sometimes when we unleash another species here to do what we want it to, it won't do just that. It may start eating a bunch of other things and menace some of the native plants. So you may know, for example, some of the examples in Australia. There are numerous ones that we let loose species in Australia, sometimes because we thought we were helping with an already problematic species that we let loose, and then we had two problems on our hands. So nowadays what's being done in terms of the introduction of these potentially useful herbivores is we screen them exceedingly carefully against a whole battery of native plants, mostly related to the natural host of this, to see if they will switch their diet. But to bring this full circle, one of the reasons why our native herbivores do not attack invasives actually makes a lot of sense when you think about evolutionary biology. Our natives have lived together with other natives in this environment, and they have learned to benefit from them, use them, or they've learned to interact with them in other ways, or learn to avoid them. But when something that's totally unknown to them, it's foreign, it's exotic, is introduced, they just haven't been exposed to it. They don't see it, so to speak. They don't detect it. And studies have been done, not enough, but the few studies that have been done have shown this time and again. Native caterpillars, for example. There are 534 species of native caterpillars that feed on our native oaks. But you introduce some other plants, and you might get one caterpillar or two caterpillar feeding on it. I'm referring to the work of Dr. Douglas Tallamy of the University of Delaware, who has specialized in just this field. So that's a long-winded answer, but I hope I answered your question. Okay. Yes?
Do you ever have trouble with your data through efforts of other organizations, either to remove invasive species or plant native species? Does that ever throw your data off?
Not on the Intertidal Zone, because we're some of the few that are crazy enough to actually go out there. So, in a way, that's a good thing. It's not the easiest access. Most people don't want to get muddy and wet. So we don't have that happening. But it's funny you would ask that. I had someone ask me that question just last week, because more and more with the threats of climate change looming, we're thinking we have to move things north to help them. That's pretty controversial. I'm not going to weigh in on that right now, but there have been people doing things like this. And with the rise in native plant gardening, there's the possibility of that. But in this project, no, I have not faced that. Yes?
Is there any chance of renaming that species that was misnamed, or because Linnaeus did it himself, it's sacrosanct?
It's not because Linnaeus did it, but we have an international body of nomenclature. It's essentially law that an international community, all of us botanical scientists, agree upon. And that governs how we name. It's called the International Code of Nomenclature for Fungi and Plants. And what that says, one of the rules is, with a few exceptions that would take me a while to explain, but very few. Once a species is named, anything that comes later, that doesn't stand. It's the principle of priority. So many times in the past, we've had one species that was named by someone in Germany and named a decade or two later by someone in France. Same species. The first one, the first name is the one that must stand. So in this case, no, we can't rename it because that name is stuck. Misnomers and all. So we have some interesting cases like that. Our common milkweed that is native to this area is called Asclepias syriaca from Syria. Similar deal. Linnaeus thought the specimen came from Syria. But the misnomers are a vanishingly small minority. Most scientific names are actually very descriptive, and they can help us remember the name for the organism. That's a great question.
Did you speak to, has there been any talk of reintroduction at all, or?
Not yet, but I would like to begin that conversation once we complete our study and have it done in an appropriate way. If we're going to reintroduce species that are here, we should use local genetic stock, like the Isoetes riparian, the river quillwort. We have those few plants left. Well, it might be appropriate to take some material for propagation from those species and then introduce them to other areas on the Hudson. Now, what do you do with something that's extirpated, like the Parker's pipewort? Well, what I would argue is go to populations that are relatively close. And some of the last remaining healthy populations are in southern New Jersey, not too far away. But even that is just the beginnings of a dialogue we need to have, we as a scientific community. Great question. Camilla?
For those species that you can only find one or two samples, do you focus more on their genetic
also have their ecological service. Not only the native species can buffer the storm.
You have some great points there. First, the genetic resources. You're way beyond where I am. I think that's great. You're thinking of the future, but I have not even begun to engage people who could test the DNA to get an assessment of genetic diversity within these species. I would love to have that. I'm hoping that we will be able to do that in the future. That's a future thing. No one ever has looked at genetic diversity within any of these species. They're so obscure, they're just not on people's radar screen. And then your other point about ecosystem services, your point that even invasive species can confer ecosystem services, I agree. But, huge but, you probably expect me to say this, ecosystem services are relative. Some are greater than others. Yes, invasive species can be beneficial in certain circumstances, like that Phragmites will buffer shorelines. But when you weigh the total package of costs and benefits of these species, in all the cases that have been studied in a local area, the invasives end up costing us in the overall cost-benefit analysis. They cost us. The harm that is wrought by Phragmites far exceeds the local benefits. So I have to be honest about it. We can reap some benefits within a short period of time and on a very localized basis. But given the total destruction, or I should say negatives, given the total set of negatives from these invasives, we're better off without them. Problem is what to do about them once they're common and widespread. Great question, great point. Yes?
So, you touched on climate change earlier on, just something that specific, really like global warming, now that we raise water. How does that affect the plants in the intertidal zone? Would that make it worse, or would, I don't know, how new species come about from the weather change?
It's another wonderful question. First thing is we have no idea. No one's begun to study that. We have a lot of people who are speculating. So I made one speculation, which is I expect with sea level rise, the saltwater front will be much farther north than it is currently. So the real question becomes, can these species migrate? Well, one thing is we have that dam in Troy. They can't migrate north of the dam. And one of the things is even north of the dam, there are highlands that stop the river from being tidal. So they have a limit on how far they can migrate. So that's a problem. But if they can migrate, how quickly? It may not be as fast as we would like them to. So with climate change, we can expect some grim scenarios. You raise the possibility, can the species somehow adapt or evolve quickly enough? Well, when you realize that the kind of climate change that we're seeing already is far more rapid than we've seen in the past, we're concerned. So in the past, we've had some tremendous climate change events like glaciations. We had, in this area, as recently as 13,000 years ago, 1 mile thick of ice above us where we're standing right here. No plants survived. Yet they recolonize and we see the rich diversity all around us right now. That was 13,000 years ago. That's not the span of decades or maybe a century like we're seeing with climate change now. So we have a lot of reasons for being concerned. We don't have to go overboard, but we should be concerned, we should be studying it. We should be asking those questions like you just asked. But I'm sorry I can't provide firm answers for it. I love all these questions. It shows that you were interested, you paid attention, you were engaged. So thank you for those. And I'm happy to stick around if any of you would like to chat a little bit or ask other questions.